Hi guys, welcome back to another video. So this one's going to be kind of a continuation of the previous kind of cells and organelles video. And in this one we're going to kind of talk about everything that I left out in that previous one because that one mostly focused on organelles. And this is kind of everything else structure-wise in a cell. So yeah, let's get into it. So first we're going to talk about the cytoskeleton. And so what is the cytoskeleton? The answer to that is that it's a network of fibers throughout a cell's cytoplasm. That doesn't really tell you much, but the purpose is to support and anchor various organelles and, kind of elements within the cell, right? So they're not just floating around. And it also helps with motility. So if you look over at this picture right here, what we've got here, this little blob, this is called a motor protein, and they've got these little legs right here, and then basically it takes an ATP to detach one of the legs and then switch it over to the front. And it walks stuff like vesicles and stuff over your cytoskeleton. So your cytoskeleton's kind of like a highway for these motor proteins, you know? So yeah, these are just some pictures of that. And then there's three kind of major types of cytoskeletal elements. There's microtubules, intermediate filaments, and microfilaments. And those are in the order of kind of thickest to thinnest. All right, so let's talk about microtubules first. They're about 25 nanometers across, and the lumen, which is kind of like the hole inside, since it's a hollow tube, is about 15 nanometers across. So microtubules are made from tubulin proteins, and each tubulin is a dimer, which means, it's made of two things, of an alpha tubulin and a beta tubulin. And they have something called plus and minus ends. So basically, when you've got a microtubule and you've got your tube, one end is going to grow faster than the other one, and that's the one we're going to call the plus end. So this kind of comes in handy later when we're kind of analyzing some things. So the purpose of microtubules is cell support and shaping, and they also serve as tracks for motor proteins, and that in turn helps guide vesicles. And they also help with the separation of chromosomes during mitosis and meiosis. So I'll be making a video on that, sometime. But basically, you can see that it's pretty important within the cell, right? So here's some more stuff. So we have these things called centrosomes, and in them we've got these things called centrioles, and you only find this in animals. It looks kind of like this. But basically, you've got lots of microtubules growing from a centrosome in your cell, and the centrosome kind of organizes all of those microtubules. And if you look at the structure of the centriole, this is something worth remembering. You've got 9 triplets of microtubules. So you got each of these is like 3 of them stuck together. You've got 9 of them in a circle, and you got nothing in the middle. It's hollow. So we call this a 9 plus 3 triplet structure. So yeah, that's something worth remembering. But basically, these are only in animals. You don't find them in plants. Plants have like kind of a different mechanism to organize their microtubules. Next we have cilia and flagella. And in these we find 9 plus 2 doublets. So how that differs from the centriole structure is instead of having 3 of them stacked like this and then put it in a circle, you've only got 2 of them in each little unit right here, but you still got 9 of them in a circle. And you've also got 2 of those doublets in the middle. So that's kind of what cilia and flagella look like. Now, there is an exception for cilia. So let's first talk about kind of how cilia and flagella are different. So cilia are shorter, and they have these things called power and recovery strokes. So this is a cilia. So a power stroke is where it kind of makes a stroke that's very powerful, and it's also got a recovery stroke to return to that original position it started the power stroke in. And this one's kind of weak. It just kind of flops back, right? It's not using a lot of force. So you can think about it like rowing. You've got the stroke, at the beginning that's really strong, and then you kind of bring your oar out back to the beginning. It's like the same thing. Whereas flagella, they have like an undulating motion, so it's kind of like a fishtail. So they move differently, right? And then for cilia in particular, there's motile and primary cilia. So when we said that the cilia were 9 plus 2 doublets, we're describing motile cilia right there. And then, this is kind of a description of motile cilia as well. But each cell has one primary cilium, and that's a 9 plus 0 doublet. So, again, that's kind of a distinction worth remembering. But primary cilia are pretty important for communication between cells, and you can kind of see that in primary ciliary dyskinesia, where your primary cilia don't move the way they're supposed to. And in development, this can cause situs inversus, which is basically when all of your organs are flipped backwards, because the way your primary cilia moves it helps your body determine when you're, a growing embryo, where your organs are supposed to be. So, yeah. And then last thing is there's these things called basal bodies, and these are 9 plus 0 triplets. It's very much like this. You find them on the base of flagella for eukaryotic cells. And and then something worth noting is that bacterial flagella, even though we call them flagella, they're like something way different. They're made mostly of protein, and they've got like 3 parts, like the rod, the hook, and the filament. That's something way different. So yeah. So here's just a little bit more about microtubules. So you've got these motor proteins, dyneins and kinesin. So you notice this minus and plus end. That basically means kinesin, it moves towards the plus end of our microtubule, so it moves towards the end that's growing faster, whereas dyneins, they move the other way. And kinesins are kind of special for this because I believe they're the only motor protein that move this way. But yeah, dyneins are also found in certain microtubule structures to bend them. So you see these red things are dyneins, and essentially when they kind of walk along one of the microtubules and a pair of microtubules, it bends this whole thing. And of course they use ATP to do this. So yeah, motor proteins are pretty important for multiple functions. Alright, so moving on to intermediate filaments, which are a little bit thinner at 9 to 12 nanometers across. You only find these in some animals, and these are good for bearing tension, which means like being squished. So microtubules, they're better at like resisting being pulled. So, you got some balance there. Oh wait, no, I think I got that backwards. But yeah, they're made of proteins, including keratin, and they're more permanent than microtubules and microfilaments. So like you see this in dead skin. I think, it's pretty common knowledge that you've got keratin in your hair and your skin, and when your dead skin falls off, like there's still lots of keratin in there, right? Even though the skin cells are dead, you still got that keratin. And you also find it in the nuclear lamina. So yeah, this is kind of what it looks like. It's basically like a coil of keratins. So yeah. Alright, now let's talk about microfilaments, or they're also called actin filaments because they're made out of actin. Specifically, it's 2 chains of actin that are twisted together, and they can branch, and they're also great at bearing tension like intermediate filaments. And cortical microfilaments help maintain the cell's shape and create the cortex. So this is like right under the plasma membrane. You've got this layer that's slightly more viscous because you've got these microfilaments that give it a little bit more structure. So this is really good for maintaining kind of like the structural integrity of your cell. And they also make up the cores of microvilli in your intestinal cells. So if you see here, this is a microvillus, and then you've got some actin in the middle. So yeah, that's kind of a nice mem to know. And lastly, maybe this is the most important function you hear about, but you have actin in myosin, which is a motor protein, and this helps with muscle contraction. So basically, you've got your actin filament, and it's kind of fixed, And myosin filaments, they're going to like walk along the actin essentially, and that helps create muscle contractions. And this also supports amoeboid motion by amoebas via pseudopodia, which are basically, that's like Latin for fake feet. Basically, when amoebas, they kind of stick out an appendage, and then, that's how they quote unquote walk. And then it also helps with cytoplasmic streaming in plants, just basically when plants move their cytoplasm around to help kind of distribute nutrients and whatnot faster. All right, so our second kind of portion of this lesson is talking about extracellular stuff. So just kind of what do you have outside your cell? So in plants and fungi, we got cell wall. So I'll mainly be talking about plants here. But these cell walls are going to be pretty thick. There's 0.1 to several micrometers, which is really rather large. And you can see kind of here's a picture, here's like a drawn picture, here's like a microscope picture. And so let's just look at how these are deposited. So basically, all plant cells have a primary cell wall, which is this orange layer right here. It's thin, relatively, not obtrusive, I guess. And then some plants, they choose to secrete a secondary cell wall. This is in between the cell membrane and the primary cell wall. And this is often lignified. It's really hard. This is how we get wood. And a lot of the time, after that secondary cell wall is laid down, the plant cell dies. So you've just got dead cells right there. Yeah. And let's see. Cellulose synthase makes cellulose microfibrils, which are embedded in the ground substance, which is a matrix of polysaccharides and proteins. So yeah, that's how your cell walls are made. You've got kind of those polysaccharides and proteins there. And then you've got those cellulose microfibrils, which are kind of like strings of cellulose, and they're laid in a specific pattern. So your cell walls are really strong. And lastly, between the cells, you got this thing called middle lamella. And this is where you're going to find a lot of pectins, which is the stuff that makes like jam and jelly have that like texture, So, yeah. All right, so that was in like plants and fungi, but let's talk about the extracellular matrix now. So this is what we find in animals. So basically you got some glycoproteins and carbohydrate containing molecules, and these are secreted by the cells, that are in this extracellular matrix. And you got a lot of collagen. So in this picture, it's these kind of light purple banded kind of long things. And collagen is really, really plentiful. It makes up 40% of total human protein. So if you think about it, that's kind of crazy. And then you've also got these things called proteoglycans, which look kind of like a feather. So you've got a protein core and a carbohydrate attachment. So it's each of these like small things right here. They can form complexes where they kind of gather on a polysaccharide. This is what they look like. So yeah, this is what a complex looks like. And then you've got these things called fibronectins, which kind of can connect your cell with this extracellular matrix. So yeah, that's basically what they do. And then they're often connected to these things called integrins, which are proteins that basically go through the membrane. And they transmit signals from the extracellular matrix into the cell. So they're pretty important. And then lastly, you should just notice that they're kind of associating with these microfilaments right here. And if you remember, that's because you've got your cortex right under, your plasma membrane in your cell, where you've got a lot of actin filaments, and that makes that whole area a little bit more solid almost. All right, so that's it for kind of extracellular stuff. Now we'll talk about cell junctions. So kind of like one classic cell junction is plasmodesmata in plants. You should know this one for sure. It allows water, small solutes, and even certain proteins and RNA to pass through. So that's how some plant viruses work. Actually, they use the plasmodesmata, and then it's big enough for their RNA to fit through and infect the plant. Also, these things, they're not just really diffusing through. You've got cytoskeleton going through the plasmodesmata, and a lot of those things are moving along the cytoskeleton. Alright, so now let's talk about cell junctions in animals. You can see we've got a couple right here. So let's just go along with the picture at first. So first you've got tight junctions, which, as the name suggests, they're really good at making a seal. And you really want these in places like, your intestines, because you don't want those leaking out, right? And also your skin. So you'll find this in most epithelial tissue, which is basically, tissue that's lining the surface of something. So yeah, hopefully that makes sense. So, it's just proteins making a very tight seal. Next, you've got desmosomes. So desmosomes are really good at anchoring sheets together. So in this case, that would just be the edges of your cells, right? And they've got intermediate filaments that are going through, like this. And that makes it really, really tight because you've got these continuous intermediate filaments going through. And oftentimes when you tear a muscle, you're tearing a desmosome, so ouch. And then gap junctions, which are like the plasmodesmata of animals. Obviously, animals don't have cell walls, so you don't have that. But you've got these proteins, kind of like these pore proteins, and that forms like a junction between the plasma membrane, and you can just have things go through. So right here. And yeah, and then the last thing I wanted to mention isn't in this picture, but they're called adherens junctions, and they use these proteins called cadherins to join the actin filaments of neighboring cells together. So it's basically a desmosome, but with actin. And they are notably weaker than desmosomes, because like we mentioned, keratin is incredibly strong. So yeah. Alright, so this is the last section, but we're going to talk about cell membranes a bit. So we've got this thing called the fluid mosaic model, and it basically states that the plasma membrane is a mosaic of proteins in a fluid bilayer of amphipathic phospholipids. So we can break each part down kind of. Amphipathic, that just means, the phospholipids, they like water and they like, nonpolar things. So that's what that means. And then, when we're talking about a mosaic of proteins, that means there's kind of like a diversity of protein, within our plasma membrane. It's not quite homogeneous. That brings us to this concept of lipid rafts, which is basically when you have kind of chunks, like kind of patches, islands of, associated lipids and proteins that are good for a specific function, and they're kind of stuck together within the membrane because that makes things more efficient. And then let's move on to the idea of a fluid bilayer. So what's interesting is there's these enzymes called flippase, floppase, and scramblase. Just kind of fitting. What they do is they essentially swap phospholipids from one layer to the other leaflet. So, they can swap phospholipids back and forth. You don't need to know this, but flippase basically swaps one from the outside to the inside. Floppase does the opposite. And scramblase, I believe, can do both. But, yeah. And then, when we mention that it's a fluid bilayer, that means it's moving a lot. And then the way it moves is actually affected by temperature. So, when you've got less saturated phospholipids, you have a more fluid bilayer. And hopefully this makes sense, because when you have less saturation, as you can see in this picture, your lipids aren't going to be able to pack as tightly. So they're going to be very loose, and that just results in them kind of flowing very easily. Whereas if it's more saturated, you're gonna be able to pack those lipids tighter together so they don't move quite as easily. And then cholesterol within the membrane kind of helps reduce the effects of both hot and cold. So kind of when it's too hot, it reduces all that fluidity. It kind of slows everything down. But when it's too cold, it prevents the phospholipids from packing too tightly with each other. And then this is just something else that you should kind of know. So there was this experiment where basically scientists, they took a mouse cell and a human cell, and they've got different membrane proteins, obviously. They fused the cells, and then after an hour, they realized that all the proteins were mixed up in that new fused cell. So that just kind of shows that you indeed have a very fluid plasma membrane. So here's just some more. Basically, when it comes to proteins, they're pretty important in your plasma membrane. You got kind of a big variety of them. So two big categories we can use are integral and peripheral proteins. So integral proteins are going to be, like stuck in the membrane, while peripheral ones, they're going to be kind of like hanging off the side. So transmembrane proteins are a type of integral protein that goes through the membrane. And something to note is that they usually have nonpolar amino acids, which are coiled into alpha helices. And an example of a transmembrane protein would be an aquaporin. And and then the peripheral proteins, they can be held in place by the extracellular matrix or by the cytoskeleton. So yeah, here's just a picture showing kind of like the different functions of proteins within a plasma membrane. You got transport, enzymatic activity, and signal transduction, which we'll be talking about very soon. And then also cell-to-cell recognition. So what's interesting about this is actually kind of how blood types work and why you can't, get a blood transfusion from like anyone, right? Because you've got these glycoproteins on your cells, and then there's a lot of like recognition with that, and then your body should not be able to find anything it should be able to recognize the self from the non-self. So it shouldn't be attacking your own cells, it should be attacking like foreign things, right? But kind of like the issue is when you get a blood transfusion, if the blood cells there have the wrong glycoprotein on the surface and that's not compatible with yours, you can have a lot of issues because your body wants to reject it. So yeah, and then you've also got some glycolipids within your membrane as well, but glycoproteins are going to be kind of more numerous. And then intercellular joining, so basically proteins in plasma membranes can help join the membranes together. And of course they can help attach to the extracellular matrix, which we already saw. And then here's just kind of talking about how proteins are going to be, really helpful for cell-to-cell recognition. So this one uses HIV as an example. Basically, HIV, it needs one kind of like main receptor, which is a CD4 receptor on a T cell, and there's also a co-receptor it needs, which is called CCR5. And basically, without both of these, HIV can't get in. So there's some people with a mutation where they don't have the co-receptor, and that means they're basically immune to HIV. So it just kind of shows how these membrane proteins can be really important. All right, so that's it for the lesson. I really hope this was helpful and you learned something. Keep an eye out for some, cell-related questions coming out soon. And yeah, thanks for watching. Please leave a like or a comment if this helped you, and please subscribe for more content.